fine for me.
All right, great. All right, so hello everyone. First, I want to thank the organizers for this opportunity to talk to you today. My name is Josephine LoRicco, and I'm a postdoc in the lab of Philippe Auger. And today I'm going to talk to you about some of my work that I've been doing, studying archaea-like membranes and how they adapt to high temperature and pressure conditions. All right, so archaea are the third domain of life, which are probably best known for their ability to inhabit some of the most extreme conditions on Earth, such as deep sea hydrothermal vents. And what I'm interested in particular is the ability of the membranes of these archaea to adapt to some of these extreme conditions. Interestingly, the lipids that we see for archaea are unique compared to those of bacteria and eukaryotes. So typically what you see when you think of a phospholipid, you see a straight acyl chain, which is connected to the glycerol backbone by an ester linkage here, and then some polar head group. And in archaea, we see that they're quite a bit different actually, where you have these branched isoprenoid-based chains here, which are then linked to the glycerol via an ether linkage rather than an ester linkage. And it's thought perhaps that some of these differences in the types of lipids that they have is one of the reasons that archaea are able to inhabit some of these extreme environments. In archaea, we also see oftentimes that they're able to produce these tetraether lipids, which are capable of forming a membrane monolayer. And the presence of large quantities of these tetraether lipids has been correlated with some of these extreme environments, such as temperature. However, a lot of archaea have only small amounts, if any, tetraether lipids. So there must be some other mechanism by which archaea with these bilayer forming diether lipids are able to adapt to high pressure and high temperature conditions. And our hypothesis is that these organisms are able to use apolar lipids, which sit at the midplane of the bilayer, and this is able to modulate the properties of these lipid bilayers. So the model membrane that we're using, we use these commercially available archaea-like lipids, which have two different polar head groups here. We have a phosphocholene head group and a phosphoethanolamine head group. And the model apolar lipid that we're going to be using is squalene. So the first thing that we wanted to do was characterize the structure of these bilayers, and we did this using neutron diffraction. So what we did is we formed a stack of bilayers with our model lipid system, and then we were able to determine different structural parameters using neutron diffraction. The reason that we are able to see some of these structures is the fact that we have this stack of bilayers, so essentially we're forming a one-dimensional crystal, which allows us to get information about the membrane structure. And the reason we decided to use neutron diffraction rather than, say, X-ray diffraction is the fact that when we use neutrons, we're able to tell the difference between hydrogen and deuterium atoms. And this is useful, for example, to determine the localization of squalene within the membrane. So this graph on the right here is showing the scattering length density of the membrane. If we look at 0 here, this corresponds to the center of the bilayer, and the minima of these plots here correspond to the polar head groups. So we have two samples here, one containing hydrogenated squalene shown in black, and one containing deuterated squalene shown in red. And you can see there's a distinct difference between these two samples, mostly around 0, which is that midplane of the bilayer. And this is how we confirm that, yes, the squalene is indeed sitting at the midplane of our membrane bilayer. So this is generally the kind of data that we get from a neutron diffraction experiment. We can see these intense peaks in the data, which are related to the fact that we have this stack of membrane bilayers. In this case here, this membrane is containing only our polar lipids, and there's no squalene in the membrane. And we see a set of diffraction peaks that are evenly spaced, telling us that we indeed have bilayer structure in our membrane. If we increase the amount of squalene in our membrane, we start to see that we can actually get a second set of diffraction peaks. So we're getting a new phase in our membrane system. Now this phase, we can see a similar phase appear also if we increase the temperature quite a bit, which is why I've named this phase phase HT for phase high temperature, whereas the original phase that we saw is only present at low temperature, so it's phase LT. So what do I mean by two phases? Well, we have two distinct membrane structures that are both coexisting in our membrane, and they differ in several respects, both with respect to the thickness of the bilayer and also of the associated water layer. Now, of course, because this is a high pressure conference, we want to see what's happening to our membrane as a function of pressure. So what I have plotted over here is we're looking at the intensity of those diffraction peaks as a function of pressure for both of the two phases. So this is the low temperature phase, and this is our high temperature phase. As we increase the pressure, going from black to green here, we can see an increase in the intensity of our low temperature phase. So the high pressure is really stabilizing our low temperature phase here. And at the same time, we see a decrease in the intensity of our high temperature phase peak. So our high temperature phase is not as stable at high pressure. So this is interesting because we can really see the contrasting effects of both temperature and pressure on our membrane structure. So we looked at the behavior of the membrane under a wide range of temperature and pressure conditions, and we're able to build a phase diagram of when the membrane is mostly in the low temperature phase, when we have a mixture of the two phases, and when we have just the high temperature phase. And interestingly, you can see there's a very wide range of pressures and temperatures at which you have the stable coexistence of the two phases. And this is very interesting because it could indicate that we're getting the formation of domains in our archaea-like membrane. And domains are something that are well known to exist in bacteria and eukaryotes, but it's something that we haven't really been able to study yet in archaea. And this is suggesting that indeed archaea are also able to form these membrane domains. And also, which I haven't really shown here, if we change the amount of squalene in the membrane, you can also modulate the amount of these different phases that you see in the membrane. So next, what I wanted to show all of you is the effect not just on the membrane phases, but on each individual phase as a function of pressure. So hopefully we can see there's an increase in the despacing of the membrane, which is the thickness of the bilayer and its associated water layer as a function of pressure for both of our two phases, although it's much more obvious for our the high temperature phase here. And unfortunately, from our data, we can't get too much more information about why we see this increase. There are two possibilities. One is that we're actually increasing the bilayer thickness. And this is something that has been seen before with membranes, where as you increase the pressure, the lipid tails become more tightly packed, and that leads to an increase in the thickness of the bilayer. And you could also imagine that as we increase the pressure, more water is being forced in between the bilayers, so we get an increase in the water layer thickness as well. All right, so hopefully I have a little bit of time left to talk to you about some of the more recent results that I found, where we tried to look at the dynamics of the membrane as well as the structure. And we did this using a technique called neutron spin echo. And instead of forming bilayer stacks, in this case, we formed vesicles from our lipid system, either in the presence of squalene or without squalene. And interestingly, so in order to form the samples for this type of experiment, we want unilamellar vesicles. So what we do is we take our solution of liposomes and we pass it through a filter with a pore of defined size to try to get uniform distribution of our vesicles in the solution. And interestingly, we saw a difference in the size between the membranes without squalene, which had a radius of about 60 nanometers, and those with squalene, which had a much larger radius. So then we took these samples and we performed our neutron spin echo experiment, which is able to measure very small changes in the energy of the neutrons that are interacting with our sample. And this can give us information about dynamics. And specifically what I was looking at is the membrane bending rigidity. So at the energy and time range that we're looking at, the the major motion that we're looking
お
at
母
in
さんの
the
教
membrane
育が聞いたんじゃ
is
ない
these undulations
ギャー
within
ギャー
the
ギャーギャー
membrane.
ギャーギャー言うから。
So if we have a very flexible membrane, we can get large undulations, and in a more rigid membrane, we see much smaller undulations. So what happens as we increase pressure, you can see for both of our samples, we get a distinct increase in the rigidity of the membrane as we increase pressure. And one thing I wanted to point out is that for these two samples, it looks like we see a difference in rigidity between the one with and without squalene, but this is most likely due to the difference in the size of the vesicles and is not actually a significant difference. Okay, so with that, I would like to conclude. So one of the interesting things that we saw in the system was this coexistence of two different membrane phases, which could support the idea that archaea are able to form membrane domains and that the formation of these domains can be regulated by squalene. We also saw differences in the both structural and dynamic properties of these membranes as a function of pressure. So it's really important for archaea to be able to modulate the properties of the membrane in response to pressure, for example, by adjusting the concentration of squalene in the membrane in order to adapt to changing environmental conditions. So with that, I will take any questions, and you can also contact me. I have my email here because I'd love to hear from you. All right, thank you.